Hi, my name is Shad Abdul Kareem, and I'm going to be doing my presentation on Janet Neisewander. Some background information on her: she got her Bachelor's of Science in Biology and Psychology and her Master's of Science in Psychology, Behavioral, and Neural Studies program, and a PhD in the same program from the University of Kentucky, Lexington. She was an assistant professor, an associate professor, and has been a professor in the Department of Psychology. She's been a professor in the School of Life Sciences since 2009. She has received many awards, which include the Bernice Grafstein Award by the Society for Neuroscience, Career Development Awards in the Basic Behavioral and Social Sciences, and Behavioral Science Track Award for Research Transition. She's a behavioral neuroscientist who uses animal models to study mechanisms of drug abuse. Her work focuses on cocaine and nicotine, which are both stimulant drugs. Her work examines social and environmental influences on initiation of drugs self-administration, the development of drug-taking habits, and motivation to seek drugs after prolonged periods of abstinence. Her research contributes to understanding the development and treatment of drug addiction. It provides new knowledge of the brain mechanisms involved in emotional processing and learning and memory. The research we'll be looking at today is titled Social Context Has Different Effects on Acquisition of Nicotine Self-Administration in Male and Female Rats. So, as an introduction to the research, the question that is asked by the researchers is, what's the effect of social context on acquisition of nicotine self-administration? Their predictions include drug intake will be facilitated by the presence of a social partner that has prior experience performing the necessary response for drug reinforcement, and females and younger animals are more likely to intake the drug. They hypothesized that social context facilitates acquisition of nicotine self-administration through reward interaction. Smoking typically begins during adolescence or early adulthood in a social context, yet the role of social context in animal models is poorly understood. The research objective is to examine the effect of social context on acquisition of nicotine self-administration. The same procedure was used for all three of these experiments. Male and female rats were housed in a climate-controlled colony. In experiments 1 and 2, they examined nicotine and it's self-administered in male and female rats. In experiment 3, it examined saline and it's self-administered in male rats. In experiment 4, both female and male rats are included in order to directly examine sex differences. In this table, you can see the experiments 1 through 4 and the sexes included in each experiment. This is a summary of the experimental designs and the conditions. These are the methods for the surgery that was performed on the rats, and these are the methods for the habituation procedures, and these are the methods for the self-administration procedures. As for the results, in summary, they found transient social facilitation of low-dose nicotine self-administration in males during the first session. However, across training overall, they found that social suppression of nicotine intake that was most prominent in females during later sessions. Subsequent tests of simple effects revealed that in males given access to nicotine experiment 1, intake was higher in the social group compared to the iso group during the first session. In females given access to nicotine experiment 2, intake was similar in SOC and ISO groups initially. However, intake was lower in the SOC group than in the ISO group during sessions 8 and 9. In males given access to saline experiment 3, the lack of a social condition main effect interaction indicates there was no difference between ISO and SOC groups. In experiment 4, male SOC rats self-administered more nicotine at the dose 0.015, which is relative to their ISO counterparts, replicating the social enhancement found in experiment 1. This image shows a timeline of experimental procedures across P and Ds in experiments 1 through 3. B is for experiment 1, C is for experiment 2, and D is for experiment 3. In this image is a timeline of experimental procedures across P and Ds in experiment 4. In conclusion, the results show that at the age of transition from adolescence to adulthood, a social context enhances the initial reinforcing effects of nicotine in males, but protects against nicotine intake during later sessions, especially in females. These findings highlight the importance of sex and social context in studying neural mechanisms involved in initiation of nicotine use. These are my works cited. And that concludes my presentation.